the characteristic and difference between these two different populations or so on. And so this is the result of phase 1 that focused on the children gut microbiota. And actually we collected about 30 to 40 children from 1 city. We actually investigated the children living in 10 cities from 5 countries. So each country gave us the sample, the children's sample from 2 cities. Two cities. One locate the urban side, and the other one located in the countryside. And so this is a result of the family level of microbial composition. And so you may simply feel that all of these microbial compositions data contained the high level population of the bifidobacteria colored by yellow, especially the children living in the East Asia are highly colonized bifidobacteria. Okay. So as a difference, so that this is a United States children at the same age, but you can't see yellow. So because they very lacking in and also the children living in the East Asia are also highly colonized by orange, that is the Bacteroides. But instead, in Southeast Asian children, including the Indonesian children, are highly colonized by the green bacteria, that is Prepotera. Okay, so it is really clear that the Southeast Asian children and East Asian children have distinctive microbial community. And this slide shows more clearly the difference. So the 303 microbial composition data at the bar graph, and I arranged the 303 data in the order of the Prepotera abundance. So left, very short in Prepotera. Prepotera is green, and right is rich in Prepotera. So you can simply see it's a quite different community. So right side, one side, right side, children microbiota, are colored by the Prepotera, the colored by green. Instead, the two side, left side children are colored by the orange and yellow. Orange is Bacteroides and the bifidobacteria. Okay. So actually, we can differentiate these two community into the different type, and the right type, so rich in the Prepotera, so we term type is bifido and Bacteroides type, so BB type, okay. And So, and this map show the distribution of these two types. So BB type colored by orange and P type colored by green. And yeah, you can clearly see that children living in the East Asia, China, Japan, and Taiwan. So they majority of they then colonized by the BB type, okay, and the children in the Southeast country, Asian countries are mainly colonized by P type, except for the children in Bangkok. Okay. And so, but yeah, as long as we only see the 16S rRNA data, we never get the functionality of these microbial community. We just know the name of the bacteria present there. So in order to address the functionality of these microbiota, so we could simply do the shotgun metagenomics analysis. The methodology is simple because just sequence everything, not only 16S, but also all of the other genes. So I can say the methodology is simple, but it costs too much, maybe more than 10,000 times of just sequencing 16S RNA. So, but yeah, but one of the Japanese scholarship can support. We got the abundance data of each enzyme encoded in each type of the microbiota. And so green means green comes from the P type, and orange comes from the BB. So in a P type, that is amylase and pectinase. Imagine that these are the enzymes which digest the plant polysaccharide. So this simply is telling us the gut microbial community of P enterotype are more active in the digestion of plant polysaccharide. So on the other hand, the BB type gut microbiota has more abundant in the gene of involved in the bile acid digestion, such as colloidal glycine hydrolase and bile acid coenzyme A ligase, so on. And so bile acid is a digestive bile acid is digest, which is involved in the absorption of the dietary lipid. So we can simply understand that the gut micro communities are very BB type are very active, more active, favors the vegetarian and DV type flavors cannibalians meat eater. So actually we get much more information imprinted in the gut microbiota of children living in India. So this is the result of a kind of machine learning microbial composition data to the computer. And we ask the computer to make data. And so this is the result. So the computer can recognize accuracy. Okay. And so this data suggests the microbiota of these three countries' children have a distinct characteristic imprinted in the 16S rRNA sequence. Okay. But for the other countries, for example, is Thailand, colored by green, and Taiwan, colored by pink. So you can recognize the location of Thailand children between the Indonesian and Chinese. And Taiwan children localized, the Taiwan children data are localized between the Chinese and Japanese. So this is very understandable because the geographical location, Thailand is between China and Indonesia, and Taiwan and Indonesia. And also you can imagine the type of food, the Thailand food are kind of between the Indonesian and Chinese. And Taiwanese eat the food between Japanese and Chinese and so on. So probably the geographical location, especially the food, daily food, have a great influence to establish the gut microbe community in the children living in each country. And actually the Japanese children and Indonesian children are very contrasted, the future. So actually it's a Japanese, so this is the diversity. Actually the number of species detected in each children. Okay. So Japanese children has quite less number of species present in their lab. Okay. And on the other hand, Indonesian the children are very rich in the variety of species. Okay. So, but generally, the high diversity, highly diversified microbial community is good, generally said. Yes. Okay. So maybe the Indonesian can be proud of your children got microflora. But this is a goodness of the Japanese, good point of Japanese, because we have a much more abundant of bifidobacterium genera. Okay, and Enterobacteriaceae, that is the non-beneficial bacterial member. So the population of Enterobacteriaceae in Japanese children are much less compared to the other countries' children. So yeah, actually, actually we have answer why the Japanese people are highly abundant in bifidobacterium, because one of the scientists in Japan recently published a paper why the Japanese are rich in the bifidobacterium, because we are short in the enzyme of lactase. Lactase. Because you know that lactose is one of the main source for the bifidobacterium, but we Japanese cannot digest lactose. That's why the lactose, the abundance are very high in our bowel. Okay, that's why we can grow a lot of bifidobacteria in the intestine. Okay. So that is one of the reasons. Yeah. And the reason why the Japanese children are less in Enterobacteriaceae, so we never know. But we can imagine maybe the Japanese children are living in very hygiene environment that may determine the level of the Enterobacteriaceae. But we don't have a right answer at this moment. And also, this is an interesting result. So from the whole shotgun genomic sequencing data. So we Japanese are very rich in the bacteria encoding the seaweed degrading enzyme genes. Okay. This may be due to the daily dietary habit in which we eat this kind of seaweed and algae every day. So that kind of the dietary component may promote the growth of the bacteria, including these seaweed degrading enzymes. And so the conclusion, the summary of the first part. So diets imprint gut microbiome from a local to global level, such as vegetarian with Levotera enterotype, or Japanese with seaweed degrading bacteria. Okay, so let's move to the second topics. Coming from the result coming from the phase 3, that is the targeted cohort study. So actually, we targeted the cohort in order to look into the in-depth correlation between food and microbiome. So actually, we investigate the gut microbiome of children living in the Leyte Island in Philippines. And also, we investigated the gut microbiome community of children living in the urban and rural in Thailand. So first, I talk about the Philippine result. So actually, we collect a sample from children living in Leyte Island, that is 8 eighth biggest island in Philippines. And and we collect a sample from these two cities. One is Ormoc and the other one is Bai city. And Ormoc is already industrialized city. Okay. And Bai is very countryside. And so this is a result of the food dietary investigation. And so this graph shows the menu of daily food taken in each children. And if the children eat some of these diet, so we put the yellow color in this graph. And so simply you can recognize that the children in Ormoc have more variety of menu. Okay? And what kind of extra menu they have is such like ice cream, dressing, chocolate, cheese, canned food. So those are, we can say that those come from Western dietary habits or so on. And actually, for example, the children in Ormoc eat the 4 times fast food a week. So, while the Bai children still eating the regional foods. This is the green mango, and also they eat banana every day or so. So these two children have a very constructive the daily dietary food, dietary habits. And also this is a composition of macronutrients. So the children in Bai, the fat composition is 18, while the children in Ormoc fat ratio accounts for 27. So generally, the highest level to keep the health is up to 13. So the people must not take the fat more than 30%. That's recommended by level by WHO. Okay. So we can say that the children in Ormoc is very close to the maximum level of the fat intake. So, and this is the result of gut microbiome. Okay. And this is quite clear. The gut microbiome of Bai children is localized in the right side in the principal component analysis. So right side is driven by Probiotik. Okay. So we can say they have the Probiotik type microflora. On the other hand, the children living in Ormoc have the Bacteroides and Bifidobacterium driven microbiota. Okay. So that's a BB type. Okay. So it is quite surprised because the children, these two cities have this distance only the 60 kilometers far from each other, and probably the ethnic group of these two cities are mostly same. So genetically same, and the distance is only 50 kilometers, but colonized gut microflora are quite different type. Okay. So, and so what differentiates their gut microflora is actually that it's driven by food. Okay, so I calculate the ratio of the macronutrient composition, and so we analyze the correlation of the ordination of the principal component analysis of microbiome composition to the macronutrient composition. And we actually got a very strong correlation between macronutrient and microbiome coordination in the PCA plot. Actually, the fat strongly driven the BB enterotype, and carbohydrate strongly driven drive P enterotype. Okay. So, and also, so we can say the BB enterotype is recruited by the high fat consumption. Okay, and actually the people, children having the BB enterotype is more obese. Okay, compared to the children harboring the P enterotype. So it is really surprising that the daily diet, very strongly drive change and affect the gut microbiome community. Okay. So, and so this is another study performed in Thailand. So we collected the sample from Bangkok. So you know that Bangkok is a metropolitan city, and we also collect the sample of the children from Buriram city. This is a rural city, and so this locates in the east and northeast part of the Thailand, so-called the Isan region. And Isan is have their unique culture, and they keep their own traditional culture. Especially Isan food is very special in Thailand, and some of them are representative of Thai traditional diet. Maybe you know that the somtam. Somtam is the papaya salad. So this is a representative of the Thai food, but it actually comes from the Isan food. Anyway, so Isan food is rich in the vegetables. Okay. And so this study is done by the Dr. Kisuse Juma, who recently got a PhD in the lab. And so this is a dietary record. So they have a different daily diet actually. So you can see that so Bangkok children eat more meat and sugar and fat and less in vegetable. So okay, and but unexpectedly, the gut microbial composition doesn't differ so much. Yeah, only this part differ between these two children. And but as a result of the metabolomics data. So to get the more than 100 the compounds produced by the gut microbes. So by using LCMS system, and and by using the metabolomics data, we make a clustering, and so we got very clear clusters. So this cluster mainly come from the children from Buriram, Buriram. Okay, and this cluster, all of these clusters come from the Bangkok children. And okay, you see that here is a strong signature. Okay, and here is also strong signature, special in the Buriram children cluster. And these are the short chain fatty acids. Okay, and these are amino acids. So the gut, gut microbiota in Buriram children are very rich in short chain fatty acid, while those in Bangkok children are abundant in the amino acid. Okay. And generally say the short chain fatty acids are beneficial metabolites of gut microbiome. And so we can simply recognize the status of the gut environment of Buriram children are good because they produce a lot of short chain fatty acids. And so vice versa, so the children in Bangkok are quite less in the short chain fatty acid. So this may be the sign of the crisis for the Bangkok children in Thailand. And actually, we have also the data of the BMI of these two children, and it is quite different. So Bangkok children have many obese. The BMI. Many of Bangkok children BMI is more than 30 or so on, while the children in the Buriram is around 20 or so on. So maybe the dietary are different, and in response to the diet, the gut microbial community are also different, and functionality of them are also different, and they may affect a lot on the health condition of the children in the Thailand. So the conclusion of the second part is like this. So urban diets drive dynamic change in not only composition, but also functionality of gut microbiome. Okay, so let's go on to the last part, so phase 4 study, to look into the relationship between gut microbiota and metabolic diseases, such as type 2 diabetes and obesity. And so before I talk about the status of the population of obesity and type 2 diabetes in all over the world. So this is the world ranking of obesity and the type 2 diabetes. And so I put a different color for each region. So, and you can see that the light blue, this means the South Pacific Ocean, so Micronesia, Tonga, Tuvalu, and so on. And so they are really fat. So they dominate the top rank of the obesity. Okay? And following then, so the Western country, USA, Canada, Australia also, and also Middle East countries follows. And how about the Asian countries? So Asian country colored by salmon pink, okay, and use, yeah, okay, and okay, and the Asian countries ranked in the latter lowest rank, okay. So this simply say that the Asian people are slim, okay, slim, okay. But for the Mongolia is among them Mongolia is the high more of these subject there, okay. And but the Philippines, Indonesia, Japan is only 5% or so. And how about type 2 diabetes? It's some Asian countries rise up to the first half rank, okay, Malaysia, Singapore, India, China, and so on. And Mongolia still the lowest rank, lower rank, and also Thailand, Japan, and Indonesia is here. It's not so bad, yeah. But maybe you know that we are also facing the crisis of the increasing of the T2D population. And even so, Asian people are not so fat, but we are facing high risk for the T2D diabetes because the etiology are different. Because Asian population, Asian races are quite sensitive to insulin, and we have less production ability of insulin shown here. And basically, we are not so fat as shown in the previous slide. Yeah, American, the average of BMI is 28.6, but we Japanese, 23.6. So even we are not categorized in the obesity, but we are facing very high risk for the type 2 diabetes. So we have to be very, very careful. And also the dietary, the diet type also increase the risk of the T2D for the Asian people because we eat basal carbon source. Our carbon source come from rice, and rice, as you know, the high glycemic index. So and okay. So we have to be very carefully look for the correlation between diet and these metabolic disease through the gut microbiota. And so to address this notion, so we did the phase 4 study of MP in collaboration with these 5 countries. And today, I show you the Indonesian data. So actually, we collected 75 samples from Indonesian living in Jakarta, and some of them are obese, and some of them suffering from type 2 diabetes. And this is a result of the principal component analysis by using the bacterial composition data. And and I put the color according to the fasting blood glucose level. So red is very high, fasting blood glucose. And you can see that here, localized very high fasting blood glucose patient here. And also, I make the size of the plot according to the BMI. So you can simply see these are very obese people, high BMI people. Okay. And and actually the bacterial composition of these three parts quite different. So this group, with high fasting blood glucose level, are very highly colonized bacteria here. And these people are highly colonized Prevotera. And these are very highly colonized Rhombocyta. Okay. This one is quite unusual. Okay. And this cladogram simply reflects the difference between lean and obese of Yogyakarta, Indonesian. So I put the red color for the taxonomic group, highly abundant in lean group compared to obese. So you see a very wide range of taxonomic group are very decreased in the obese group. And these are Luminococcus bacteroides and Clostridiales or so on. So we can simply recognize that the gut microflora of obese group are very unusual. It's very poor in the commensal group. Okay. So this, and also that we can simply calculate the alpha diversity, the richness and diversity of the microflora. And so the obese people from Yogyakarta are very short in the bacterial richness. Okay. So very poor in the diversity of the bacterial species. So it's really unusual. So, and this kind of status of the microbiome, we simply say that this biases. Okay. However, if we compare the gut microbiome between T2D and non-T2D, okay, difference is not so much. Only the specific part of the microbiome are different. So for example, this one, okay, this is only one species, Bacteroides fragilis, are abundant in the T2D group compared to non-T2D group. So, what's happened in the T2D group? So, highly abundant of the Bacteroides is one of the typical characteristics in the T2D patient. So I I show the graph in the order of the fasting blood glucose level from left to right. So these are very high fasting blood glucose level patient. And here I show the bar graph of Bacteroides fragilis and Bacteroides colored by orange. Okay. So you can see these group, only these parts have a high abundance of Bacteroides fragilis. And the rest of the group, patient, subject doesn't have Bacteroides fragilis except for a few exceptional. So probably Bacteroides fragilis have some very bad functionality to promote the diabetes. Okay, and we also have microbiome, so metabolome data, and we found these people are highly abundant in the conjugated bile acid, okay, here. So this is the distribution of the conjugated bile acid level among non-T2D subjects here. And here is T2D subject. So you can see, yeah, the conjugated bile acid level of the T2D patient of Indonesian are very low, okay, as shown here. And this is also the actual data. So these are unconjugated bile acids, and these are very poor and compared to the rest of the subject. Okay. But okay, you can see that the subject that marked by star, red star. What are these? These had metformin. Metformin is the anti-T2D drug. Okay, so they are administered by metformin, and they know more bacteroides. Okay, here, 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 and the level of the conjugated bile acid is recovered. Okay, and there are some papers about the relationship between the conjugated bile acid and Bacteroides fragilis and type 2 diabetes. So in this paper, they did some animal experiments and also a human intervention study. And in the human study, they give the metformin to the T2D patient and to see the change in the abundance of Bacteroides fragilis and also conjugated bile acid. And they found the subject who are administered by metformin decrease the level of Bacteroides fragilis as well as conjugated bile acid recovered, okay, after intervention of the metformin. Okay. So, but this is intervention study, but our Indonesian study is natural. Yeah, we investigate the natural T2D patient. Okay. So, but we got the exactly same data as observed in the human intervention study using the Western population. So this is the summary of the microbiota-mediated bile acid metabolism and its disorder associated with T2D. Okay. So lean non-T2D patient, this is the healthy people. So the conjugated bile acid are secreted from gut from the liver, and so by the functionality of the commensal bacteria, it deconjugated, and deconjugated bile acid converted to UDCA or 7-alpha dehydroxylated bile acid. So this is the final end product of the bile acid. And it is known these bile acid is important to modulate the host receptor, that is FXR and TGR5 or so on. This receptor is very important for the control of the fasting blood glucose. Okay. So in the healthy people, this stream is going smoothly. Okay. But in the T2D patient, overgrowth of Bacteroides fragilis, and Bacteroides fragilis have very a high activity for the deconjugation. So actually, a part of the conjugated bile acid must remain here. But T2D patient with high bacterial fragilis having a bile salt hydrolase activity, it's the conjugated bile acid is depleted, okay, and no more conjugated bile acid. And conjugated bile acid also very important to control the receptor activation, okay. But the T2D patient, less conjugated bile acid have a bad effect on the receptor activation, okay, due to the loss of the conjugated bile acid. But metformin is known to block the growth of bacterial fragilis, and the level of conjugated bile acid is recovered, and this antagonist activity is recovered, and the good to control of the effects are receptor activation. So that this is one of the known mechanisms for the cure of the type 2 diabetes. And we also analyze the correlation between the food and microbiome. And finally, we found a positive correlation of fat with the lombocytor, and also positive correlation of carbohydrate to the luminococcus. Okay. So fats driven the growth of the lombocytor, and lombocytor is, it's not beneficial bacteria, so promotes the obesity. Okay. But it is quite strange because they didn't suffer from the diabetes. And actually, we found the lombocytor produced the succinate, and succinate is known to block the increase of the blood glucose level. So maybe this is one of the magic of the Indonesian obese people. They suffer from obesity, but protect the development of type 2 diabetes. Okay. But this is just my speculation, so it hasn't been proved yet. Okay. Also, let's focus on the luminococcus. Luminococcus is the commensal root, okay, and actually this is the counteract with lombocytor and Prevotera, and so it's blocked the increase of the weight, and so this may be goodness of this bacteria, but at the same time, if the bacteroides, especially the Bacteroides fragilis, increase, this promotes the increased level of the fasting blood glucose. So maybe it's time to finish. So the conclusion of the third part is that microbiome of obese patient is characterized by this dysbiosis-like feature, while T2D microbiome is locally changed with Bacteroides fragilis. Okay, but also enterotype shift from Prevotera to Bacteroides seems sensitize diabetes to people in developing countries. So finally, I would like to thank to my colleagues in phase 4. So the Indonesian sample is collected with a lot of contribution by the Professor Endam. And the analysis of the Indonesian microbiome mainly done by Mr. Patanaponte Radatada. So, who is going to get a PhD, hopefully, with this study soon. And okay, so finally, I wish the end of coronavirus pandemic, and I really hope to see you face to face in each country. Okay, so that's it. So thank you for your attention.